Thank you so much. This is an amazing moment for me. It's a huge honor, and I am incredibly humbled and grateful by this award. I would like to start by actually thanking, are my slides on? Can I have my slides on? So I would like to actually acknowledge Peter and Patricia Gruber for their vision a long time ago to support frontiers of science in cosmology, genetics, and include neuroscience and justice for women. And you don't need to see that. Okay. Justice and women's rights. I also thank the advisory board. I think the composition of the advisory board makes this honor even greater. They are all people I admire greatly, so thank you. I have been involved one way or another with the Gruber Foundation since 2004, when I joined the first actually selection board committee with these wonderful colleagues that you see highlighted here. And I remember as it was setting up the relationship with the Society for Neuroscience, the depth of involvement of Patricia Gruber in thinking through all the logistics in announcing the prize for Seymour Benzer, who was the first awardee 20 years ago, who's a pioneer in neurogenetics, and in thinking through the right way to interface between the Gruber Foundation and the Society for Neuroscience. And it's obviously proved to be extremely successful and impactful. So thank you, Pat. And I had the honor of meeting Seymour Benzer, one of my heroes, and we had discussions about the genetics of emotions, and I was just starting to think about how we could approach that in rodent models, and I got this lovely note from him after the dinner award, almost exactly 19 years ago, saying that it will be fun to seek out the similarities between rats and flies and ourselves when it comes to the neural substrates of emotional behavior. So this is sort of my topic today, and I think it's kind of a fitting recognition of his long influence and this early interaction in the context of Gruber. This honor belongs to many other people, not me, and that first and foremost includes my husband, Stan Watson, who has been my collaborator throughout and colleague and throughout all the years of our research. You can see him as his beard has gotten wider. And we have many joint trainees that we call Wakils for Watson and Akil. We love dearly. This is a list of current Wakils, and I will also touch on the work of a few past Wakils. Briefly, please forgive me if I summarize years of work in one sentence. I also want to acknowledge some collaborators that have been ongoing for a while. And I want to also mention two amazing consortia that I have been part of. One is the Pritzker Neuropsychiatric Research Consortium, and the other is the Hope for Depression Research Foundation. These two consortia with amazing scientists in other institutions, all tackling the challenge of psychiatric disorders using both basic and translational research, have really changed my view of how we need to do science these days and how productive it is to work with people with different skills, but with a common goal. So thank you all. I have no conflicts to declare, and I want to move on and talk to you a little bit about the emotional brain. My topic is embracing complexity, and I want to start by saying that for a long time, it felt to me as if affective neuroscience was the stepchild in the neuroscience field. Nobody really was studying it, or not in a very visible way. And I think that part of the reason is that there is no easy foothold for studying emotions and mood the way we have for sensory system, motor systems, learning and memory, motivation, where there is either a stimulus or a response or an effect that you can study. And yet, emotions shape all of these functions: perception, motivation, learning, memory, and action. They really are sort of the elephant in the background that nobody ever wanted to look at. And one way I think about them is that they provide salience in a very complicated world that we need to evaluate and predict. Emotions and stress are ways to tell us what is important and what we need to commit to memory. And so, in a way, therefore, I think of affective neuroscience not just in terms of occasional emotions that we feel, there is that, but there is also this whole ongoing process playing in the background that gives meaning to our experiences. So it is complex for that reason. It's there all along. But E.O. Wilson said complexity is what interests scientists, not simplicity. And so the question is, how do we unpack this system? How do we think about levels of organization, levels of analysis, how we think about time as a dimension, how we consider checks and balances that might lead to susceptibility or resilience, and how we always keep in mind the notion that not everybody is the same, that there are profound individual differences that shape who we are and how we respond to the world. So in looking at the dimensions, let's start with something simple. This is a painting by Picasso in the Blue Period. We can definitely see that the lady seems sad or down. So valence is one of the first things we can think about in relation to emotion. And this is her when she's feeling better. And always a question is, how long does this last? So is it stable or is it labile? And the issue of time defines actually many concepts in affective neuroscience. So I think of emotions as being relatively short-lived, of mood as being longer days or weeks or months, and of temperament as being lifelong tendencies. And then if we have all of these elements in place, how do we study them? Well, we can obviously go from gene to cells and circuits and behavior and even social context. But we also need to keep in mind development, which constantly reshapes these processes. So it is these elements then that I would like us to keep in mind as we proceed. Genes and development, broad environment, like where you grow up, how you grow up, but also specific experiences and individual differences in coping. And for me, a major goal is to understand well enough to predict. That doesn't mean you fully understand, but you understand well enough to predict, because if you can predict, you can start to prevent. So you'll see how well, whether I succeed or not, but I'm going to give you examples of that. So this talk today then has elements from some vignettes from animal studies, animal models, and a couple of vignettes from human studies and a synthesis at the end. And one criterion is that animal models need to be informed by the complexity seen in humans. So the idea is to simplify, but not too much. So let me start then with genetics. And we're going to talk about temperament. Temperament, as I said, is lifelong. We're looking at the gene level. We're going to also think about development. So I'm going to start by asking, what is your idea of fun? Is it to sit at home knitting, or is it to go skydiving? Probably most people are somewhere in between, but those are kind of extremes. And the reason I bring it up is because these tendencies tend to be part of personality styles that represent a continuum of emotional reactivity and ways of coping, and that have been associated with very different types of disorders called internalizing disorders versus externalizing psychiatric disorders. Internalizing disorders include depression, anxiety, suicidal behavior, and are considered over-inhibition. Externalizing disorders are conduct disorders, antisocial behavior, substance use disorders, not enough inhibition. And there is a lot of data from genetics and genetic epidemiology that says that this distinction is actually functionally and biologically relevant, that there are genes for that more likely than specifically for just depression or just anxiety. So we set out to see if we can model this. John Stead, back then in the lab, had the idea of how to breed them. We started from outbred Sprague Dawley rats. We did not want inbred lines. So it was outbred animals that we then tested on a behavior that Piazza et al. Had noted, that if you take animals and put them in a new environment that they have never seen before, they either tend to be very cautious and hug the walls, and that is here on the left, we call them the low responders, and others who love that it's new and exciting and go all over the place and explore, and they are the high responders. And you see the B because we're going to genetically breed them. So his observations were in non-bred animals. So we set out to genetically breed these animals, and you can see from how quickly they diverged that it's very genetic. They diverge, and in fact, it's very heritable. You can tell with almost 100% certainty what the phenotype of the animal is based on their family lineage. So examples, this is from Aiden et al. A very simple intermittent mild stress. If you subject the high and low responders to that, and you look at the social interactions that they have with other animals like them, their buddies, what you find is that the low responders become really inhibited if they have been stressed, whereas the high responders don't change at all. So they start at the same level, but they're differentially sensitive to stress. This is ameliorated by antidepressants. And there are many features like that where the low responders show greater negative emotions, stress reactivity, spontaneous anxiety, depressive behavior, fear conditioning, fear extinction. You can see some of the references at the bottom. By contrast, if you go to the other side and you ask about the high responders, you expose them to cocaine and other psychostimulants. And the buddies, those skydivers, love the drugs. So from the first day, they start administering at full speed ahead, whereas the low responders tend to be more timid about that unless they are stressed. And there are many behaviors that are along the same line. Shelley Flagel, in particular, has worked hard on this facet of the high and low responder phenotype, showing that every facet of cocaine addiction is actually differentiated in these animals. We have more recently shown, Michael Emery, that opioid self-administration is different, sensitization to drugs, impulsive action, novelty-seeking, sign-tracking behavior, Shelley again. So very different phenotype there. And all of these phenotypes show up early in development. So we feel that we've created an animal model or two animal models that map very nicely on human internalizing and externalizing disorders. And so the question is, can we shed light on the genetics of these temperamental tendencies? And for that, we're using a combination of genetic sequencing and gene expression profiling and behavior. This is a NIDA effort with Jun Lee and A. Palmer, and you could see some of the publications that are recently coming out. And I want to summarize a lot of work first with a sentence by saying that this is not one gene and it's not one circuit or one cell type. I feel like even though we bred for a simple behavior, somehow this breeding changed the fabric of the brain. These animals are really different from each other with different cell type balance and different interaction. Here is a more numerical way of looking at it. So this is Hebda Bauer, Hagenauer et al. And the number of genes in the hippocampus that are differentially expressed between high and low responders is about 5 times higher than the number of genes that are differentially expressed between males and females. So these are really significant, profound differences that have arisen. So in order to hone in on the ones that are potentially functionally significant, we need to put together differences in sequence, differences in gene expression, differences in behavior, both in the parents and in offspring that we have bred together to break up the haplotypes. And when you do these analyses, what you find is that there are some really interesting hits where genes that are part of the Manhattan plots where you're seeing differences at the genetic level also show differences in gene expression, EQTL, and behavioral differences, QTL, that all converge. And there are entire pathways that emerge from this analysis. They include neurodevelopment and synapse formation. They include neuroinflammation and microglial function. They include energy regulation and mitochondrial function. And one thing that I really like, and I want to thank Megan Hagenauer because she has pushed on this idea, is to look at convergence with other animal models and with humans. And a lot of our top hits, either genes or pathways, are actually shared with other animal models of affective disorder and with some human GWAS. I mentioned mitochondria. We see, for example, that the gene expression pattern is consistent with the idea that bred high responder animals have nicely controlled energy production that's responsive, whereas the low responders are running around with high oxidative phosphorylation, but maybe less responsive to cellular needs. Now, this is a hypothesis. Then we need to go then individually to some of these genes and functions and test that. But this is a hypothesis generating stage of the research. So having characterized some of these features of the high and low responsive biology, the question is this fate or can you change it? And especially during development, and for that we have tried a number of approaches. I'll give you 2 examples now and another a bit later, molecular and environmental. So one approach was done by Pam Maris in the lab, where she built on the observation that microglial function seemed to be changed and did the morphology of microglia in the high and low responder animals, and noted that even though the number of microglia was the same, their pattern of arborization was really different. So they were much more arborized in the more anxious animal, consistent with a hyperramified state that's associated with stress and inflammation, even though we had not done anything to them. But can you change that? She used a simple drug, an antibiotic minocycline, which is known to alter microglial function, in the drinking water for 2 weeks. And it did not affect the high responders at all, but in the low responders, we saw increased social interaction, decreased immobility and increased swimming and activity in the 4-swim test, and increased sucrose preference, which is a measure of positive motivation. So it alleviated anxiety-like phenotype in the low responders with the messed up microglia. Another approach is O'Connor, Hagenauer et al., where we're looking developmentally and environmentally. So high and low responder animals either live at home in the usual boring places, or they live in a more enriched with toys and other animals interacting with during adolescence between P35 and P60. And then we bring them back, and some of them get exposed to a social stress, and the other half is not. And then we test them on a number of tests, and I'm going to tell you about social interaction again. And I'm going to show you one example out of lots of data, behavioral and non-behavioral, and that is what happens after social defeat in the low responder animals. So as you might imagine, they are shy. I told you they're sensitive to stress. They become very avoidant even towards their buddies after they have been exposed to a social stressor. But if they had lived in an enriched environment before that, while they were adolescent, they are very highly protected from the effect of social stress. They are, in fact, more interactive than the genetically high responder, the genetically resilient animals. So there is a neural phenotype also associated with that. So I'm not going to say much more except to say that we believe we have a model of temperament that has a genetic basis. I hope that, though, that I've convinced you that it's not immutable, that it can be changed, especially during development, and can be targeted especially better if you know the things to target, like microglia. So I want to move on and talk about the impact of a single gene and development. And so we're going to look at stability of emotions here. We're going to look at a short period of response. We're going to look at a gene. So, and here we're going to focus on an old friend, the hypothalamic pituitary adrenal axis. This is the biology of the neuroendocrine system we know a huge amount about. A lot of the molecule and the end product is the production of glucocorticoid stress hormones. We know glucocorticoids go back to the brain and inform the brain that stress response has happened. And the glucocorticoid receptor, GR, is a sensor of that in the brain, in the hippocampus in particular, and says, stop, okay, stop freaking out, we know, okay. But while that was known for quite a while, we didn't know very much about the role of GR in emotions. So when we set out to do this work, Chang Wei created a transgenic animal in collaboration with Audrey Seschultz, where we wanted to express the glucocorticoid, overexpress the glucocorticoid receptor only in the forebrain. And you can see that the transgene is expressed in hippocampus and cortex. Chang also created an inducible model where you could turn on and off the expression of that gene. So you could see, again, you can express it here very selectively in hippocampus. And what that let you do is you could let an animal grow up normally and be a wild type, or overexpress GR, increase the glucocorticoid receptor throughout their life, or do it before they were weaned only and then let them grow up normally after that, or let them go to weaning normally and then overexpress afterwards into adulthood. And when we look at the sum total of the data, we were surprised that overexpressing GR in the brain did nothing to the endocrine system, the endocrine response, but it produced a very emotionally reactive animal. These animals were more anxious and more depressed like ordinarily, but you could swing them from that state very easily with a number of ways, including antidepressants, which is something that's seen in bipolar disorder. They were very sensitive to psychostimulants, and their gene expression patterns were extensive, and some of them also echoed what's seen in bipolar disorder. And the remarkable thing is that this entire behavioral phenotype got laid down before weaning. So if you did it before weaning, you saw the whole picture that I just described. If you start overexpressing GR after weaning, you don't see that phenotype. So it makes me, I call GR a molecular organizer because it plays this critical lifelong role in shaping emotional behavior. So more recently, we wanted to see more circuits that are associated with this high emotional reactivity. So Chang Kumar et al. Used optogenetics, both in dorsal and ventral hippocampus, looking at the elevated plus maze to see how stimulating these animals changes exploration and risk-taking behavior. And what we see is stimulating in the ventral hippocampus does increase exploration in the wild-type animal, but increases it way more in the GR overexpressing animal. So the same stimulus exactly, optogenetic stimulus, very dramatic difference in reactivity. Moreover, it lasts longer in the GROV animals. So we wanted to know what cell types were involved in this. So where in the brain and what cell type. And when you do CFOS mapping throughout the brain, you see that optogenetic stimulation changes the brain in many, many loci. And you can compare wild type and GROV, and we quantitated all of that. And the region that stands out as really overreacting in the GROV animal are ventral hippocampus and amygdala, posterior amygdala in particular. So what are the cell types in there? So this relied on the use of multiple fluorescent in situ probes. We're using, so you could see where channel rhodopsin is, FOS, glutamate, GABA, and their coincidence. And as you could see, using this method, once again, the amygdala and CA1 are showing up purple, which means they have co-expression of glutamate, V-glut, so they're glutamatergic, and CFOS. So these glutamatergic receptor cells are being overactivated in the GROV animal compared to the wild type. And you can quantitate that here painstakingly. You can see that in GABA neurons, the wild type and GROV are not different, but the glutamatergic neurons show consistent differences only in the amygdala between the two lines. And if you go further and drill down on the types of glutamatergic neuron, we've found out, again, using multiple probes, that it's calbindin-expressing neurons, specifically in ventral CA1, that are being differentially activated in those highly reactive animals. And it is DARP32 expressing neurons in basolateral amygdala, posterior basolateral, and neighboring nuclei that are being activated. And so this speaks to the existence of a pathway which had been identified before, which is a reciprocal pathway between the hippocampus, ventral hippocampus, and posterior amygdala. We know now it's GABAergic. It contains calbindin on one end and DARP32 on the other, and it seems to modulate reactivity, emotional reactivity, and this is completely consonant with earlier work by Gillen et al. Showing that this pathway is important for approach avoidance behavior, but we showed that GR changes it. So again, as a future direction, it would be important to intervene with this pathway and see if you could stabilize emotional reactivity. But what I think is cool more broadly is that a single gene could tune emotionality, and that by having knowledge of that initial genetic trigger, the cell types involved, the specific circuit involved, it makes it possible to imagine treatments that target these cell types, these genes, and see if one can stabilize emotional reactivity in certain disorders like bipolar and maybe personality disorders. So the last bit about the animals that I want to tell you about has to do with experience. So we're going to look at kind of an intermediate period of responsiveness, and we're not going to do anything genetic. We're going to look at cell and circuits and behavior. And for that, we're going to use a model that Eric Nestler and his lab have just fine-tuned beautifully. And it involves taking an inbred line, black 6 mice, and putting them with a more aggressive white mouse CD1 for 5 minutes, where the CD1 tries to attack the black 6 mouse. Then you take the black 6 mouse, you rescue it, you put it in a neighboring chamber for 24 hours where they can smell the aggressor but not ever interact with it. And you do that for 10 days in a row. And the main readout is social interaction. Dalia Mura in the lab repeated all of this and showed that there is a normal distribution in terms of how much animals like to socially interact normally, but as the Nestler lab would tell us, after social defeat, the animals break up into 2 groups that they have defined. One is a resilient group that more or less acts like nothing has happened. They still are socially interactive. And the yellow group, the susceptible group, that tends to be much more inhibited, especially here. They really don't want to interact with another animal. They would just rather avoid it altogether. So it's important to point out that, again, this is not genetic. They are the same. And that the experience takes a few days to solidify, maybe 5, I don't know, Eric can correct me. But you cannot, so the question is, can you predict? Can you predict who's going to be susceptible or resilient in the future? And we know, and many people have tried, classic anxiety and depression measures do not predict how these animals are going to emerge from that experience. Dalia tried to do much more characterization on day 1 during the fighting, and had a small but significant correlation with escape behavior. So animals that tended to want to run away tended to eventually emerge as more susceptible. But it was not just a strong predictor. So we wanted to look at the brain, but we wanted to look on day 1 because we want to predict. And we want to look at the brain and see what's happening there, but allow the animals to live on, go through the process, and emerge as either susceptible or resilient. So we needed a way to do that, and for that we used this method called fast trap that lets you do that, lets you capture neuronal activity on the first day, and then let the animal live and still see it at the end. And this is a neuron labeled with DT tomato that was active during defeat 10 days ago and now shows up as still labeled. So when you do that, you can compare groups of animals, animals that sit at home quietly, animals that are just interacting with a friendly neighbor, but not having gone through defeat, and animals that have gone through social defeat. And you could see that there is actually more fast trap activity in defeated animals. There are certain brain regions, like the medial habenula, that differentiate between susceptible and resilient animals. But our goal was to look more brain-wide. So we used NeuroInfo for a semi-automated quantification and identified 73 different brain regions. By we, I mean Dalia Morra and colleagues. And we conducted heat maps. We need correlations of fast trap activity on day one to see how it relates to the phenotype. So here is control versus defeat, and you could see that the experience of social defeat makes the animals' brains work differently. You see a lot more correlation between the activity of different brain regions in the amount of fast trap that they express. If you look at susceptible versus resilience, you see that two different patterns emerge with the future resilience. So they will be resilient. On the first day, animals that will be resilient will have a lot greater correlation in their brain activity than animals that will be susceptible. And there are certain regions that are highly correlated. In overall, the ventromedial hypothalamus, VMH, was the most correlated region with all the other 72 regions. And you could see, you can use it as a seed and map it, and you could see how it was not that correlated with the network in control conditions. It became highly correlated with an entire network of regions after social defeat. And the pattern was different between susceptible and resilient. For example, CA3 regions showed up as a highly correlated area with VMH in the future susceptible animal, but not in the future resilient animals. Moreover, there were individual regions that were predictive of the social interaction score in the future. Again, this is like 15 days later. The paraventricular zone, the cortical amygdala, posterior amygdala, intralamina nuclei, all the level of activity in them predicted future susceptibility or resilience to the stress. Optimally, you want to also look on the last day, and you want to see how the network changes over time. And we don't have any data, but the technology is in place. This is Kumar et al., where you could see FOS in green, you could see FOS trap in red, and here in yellow is a cell that has both FOS and FOS trap. That means that neuron was activated on the first day and also on the last day. So you can start to track that and see how the network evolves. But my conclusions from this is that we see enhanced coordination with social stress. We see distinct anatomical nodes and coordinated activity depending on the future reactivity of these animals, and certain specific regions are predictive. And more importantly, that there is something happening beyond genetics, which relates to experience. Maybe not a single experience, but maybe that first experience creates a bias, and with repeated exposure, it creates a very different kind of network activation in the brain of these animals that eventually leads to the different behavioral phenotype. So that also goes to the idea of prediction, not for genetic, but experiential reasons. So what have we learned from the animal studies I've told you so far? That genotype, that there is genetics to temperament, but that there is resilience also beyond genetics. That there are molecules that are uniquely powerful and act as molecular organizers during various stages of development. I gave you GR as an example. That we have identified a neural circuit associated with emotional lability. We don't know if it's causative, and we need to test. We associated a neural network that encodes the initial experience and appears to shape future emotional reactivity. And again, you need to eventually completely intervene and get in the way. But first, you have to identify. So I want to move on to talk about human and very briefly tell you about a ton of studies in a few minutes because they are a nice connection between animal and human research. And this is work on post-mortem humans. So it's on mood, so it's intermediate duration, and it goes from genetics to circuit. And it's work with the Pritzker Neuropsychiatric Consortium, where post-mortem studies are seen as an intermediary between genetics and clinical phenotype. And again, I will start with a very general statement by saying that when I look at all the data from all the gene expression studies in depression, my feeling is we can't attribute it to a few genes or a particular circuit, but it is like this tree in summer versus winter. The winter one is reminiscent to me of a depressed brain. It looks very broadly altered and impoverished in many ways. So some of the pathways that have been identified by the consortium over time are cell type balance. Again, clock genes, we have worked on them. Immune genes, growth factors. I'm going to say a few words about the growth factors. Simon Evans in the lab early on identified the fibroblast growth factor family as being highly dysregulated in depression selectively, not like, as opposed to not just controls but bipolar and schizophrenia. Some genes were downregulated, like FGF2 and its receptors, whereas others, like FGF9, were upregulated. But the question is what that meant, and for that, we needed to go to animal studies. And we looked at all the elements that we could think of that might impact depression, from vulnerability genes to development, to the role of stress, to the role of neural remodeling. We looked at the role of FGF2. This is a series of studies that Courtney Turner has been very instrumental in, and many others in the lab. And the bottom line of a lot of this work is that FGF2 is our own natural, occurring in our brain, antidepressant, anti-anxiety substance. And in our little model of the high and low responders, it's natively different. The poor low responder animals that are more anxious have less naturally FGF2 in their hippocampus. Examples of the kind of work include knocking down FGF2 specifically in the dentate gyrus, Kosak et al., Auerbach et al., and showing that that leads to an increase in anxiety. That means natively it's playing a role in anxiety behavior. And by contrast, chronic administration of FGF2 in the adult, Courtney Turner has shown that it produces both anti-anxiety and antidepressant effects. Moreover, FGF2 changes neurogenesis. This is Perez et al. And importantly, it does it only in the low responder animals that have less FGF2 to begin with. And if you expose these animals to an enriched environment, and I told you that that induces resilience in FGF2, it also induces, it induces resilience in the LRs, that it also enriched environment induces FGF2 selectively in the LRs. And if you use an antagonist, you can, in fact, block the effect of enrichment on the animals. So another kind of a really cool study, I think, by Courtney Turner was giving one shot of FGF2 on the second day of life and letting the high and low responders grow up to adulthood, testing them on the elevated plus maze. And what you could see is that they are really different at baseline. The low responders hardly ever take a risk and go on the open arm. They're very conservative. But if they've had FGF2 as babies and allowed to grow, that one shot makes them more like average animal. They're not as crazy as the high responders, but they are pretty average. That treatment also has many other consequences. So it acts like a molecular organizer. It increased neurogenesis. It produced lifelong changes in other genes. It created epigenetic changes, and behaviorally, novelty-seeking, fear extinction, and resilience to social defeat were all ameliorated in the low responder animals. So starting from depressed brains, we uncovered this factor that seems to modify vulnerability or resilience to depression and seems to be an endogenous resilience factor that can be induced by the environment, by environmental enrichment and change neurogenesis. And I'm very happy that a lot of people have replicated and extended the FGF2 studies, FGF work, under many different other models and their role in antidepressants. So my last piece is what I call real life, which is what happens to real people living in a stressful time. And that's the Michigan freshman study. So we're going to study them for a year, and we're going to look at genetic and social factors. And the premise of the study is very simple. You take healthy young people at a time when they're about to face what is likely to be a stressful experience, and you ask, how are they going to emerge? Are some people going to emerge unscathed, and other people will suffer from stress-related disorders like depression or anxiety? So to do that, we do a very complete baseline characterization of their psychological and biological profile. And then we study them longitudinally throughout the year on regular intervals, and we look at the incidence of depression or anxiety. And again, the question is, can we predict? Can we go from that initial baseline and predict what's going to happen? So we had started when the pandemic hit. So these kids then went from living a typical Michigan college life, which is all about Michigan football, yay, to having to pack up and go home for the quarantine. So this study I will tell you about has 3 phases, 3 cohorts: a pre-pandemic cohort, the pandemic cohort for year 1, 2019, 2020, and then a pandemic cohort for year 2 to 20 to 21. This is the general design where we do a whole battery of tests and mood tests, salivary cortisol tests, and so on. I need to acknowledge, again, Courtney Turner, Ginny Weinberg, Huzaifa Khalil, and other collaborators on this study. During the COVID pandemic, we had to pivot, but we actually added more mood and saliva measurements on a monthly basis. We did an endocrine profile before the pandemic where we exposed them to a formal social stress test and got their endocrine profile, but we needed to stop that during the pandemic. We just resumed it again. And we give everybody a Fitbit, and we download all their activity and sleep data throughout the year. So what did we learn? Before the pandemic, the freshman year was indeed stressful. A certain cohort, 16% of the kids developed depression, 18% developed anxiety. We had a baseline of 4, and a number of them had either and both. What happened during the pandemic was actually startling, especially where depression is concerned, where you could see that it went from 16% to 33% rate of depression in the cohort in 2020, 2021. So this is a bar graph showing the baseline in orange and then what happens during the course of the year, and you can see the steady increase in rates of depression in each of those consecutive cohorts. But what this is not showing you is the gender difference, which I think is remarkable, because it's all about the women, the young women. Pre-pandemic, it was 2 to 1, which is what we expect in females. It became 4 to 1 by the 3rd year of the pandemic. The females were really hard hit, and you could see that in a number of ways. You could see that about half of them had either depression or anxiety, clinically relevant meeting criteria for depression or anxiety sometime during the course of the freshman year. A greater percent of females had both depression and anxiety coincident, and there was greater chronicity, meaning month after month, they were still depressed and anxious. If you look at the pandemic year, the first pandemic year itself, it's actually also remarkable. So this is before and after the quarantine. And what you see is a measure of anxiety went up very dramatically and pretty clearly. Depression went up very dramatically. And movement, daily movement, dropped really spectacularly. So here is records of the Fitbit data, starting from September to the following December of their sophomore year. This is when March hit and they went home. They stopped moving altogether, but they never really bounced back during the summer, and into the fall when they came back to college as sophomores, they still were moving a whole lot less than they did a year before that. So the question is, how genetic is this? And to measure that in humans, we use the polygenic risk score, which is a global measure of genetic vulnerability to a particular condition, in this case, major depressive disorder, so it's called MDD-PRS. And before the pandemic, in fact, having a certain genetic score was predictive, especially in the females. So if you had low genetic score, your odds of developing depression were low in the year. But if you had a high genetic score, your odds were higher. But that completely disappeared during the pandemic. We no longer had a correlation of any type, males or females. So there were, for example, these females that were predicted in the subsequent cohort. There were females that had as high depression as anybody else and had very low genetic risk. But equally interesting is what happens to people with high genetic risk who do not become depressed. So that's a measure of resilience that can't be genetic. So this is an example of another group of females that never developed depression, even during the pandemic, and with a high genetic risk score. So the question is, what can we predict with anything if it's not genetic? And Jose Khalil used machine learning and applied it to all the measures that we had at baseline, all the psychiatric measures, and derived something called affect score that had a small component that related to family history, a component that related to traits, so more temperamental, like neuroticism. But it turns out that a very big component of this affect score that is predictive relates to the state of the person, the freshman, as they enter the freshman year. The state of mind at that moment, how depressed, how anxious, how suicidal, how positive, how negative. And you could see here the correlation of the affect score is pretty spectacular, I think. Like minus 16. So it is a very strong predictor of future susceptibility or resilience. And it means that there is something psychological at the start of the year that's protecting these kids from further stress in spite of their genetic vulnerability. So what happened since? We continue to monitor the kids every year, new set of freshmen. And sadly, depression has not gone down in 21-22 or 22-23. You can see it's still at about the same level, maybe a slight drop in baseline this last year. And really importantly, the affect score is very, very predictive. And remember, it was derived from a completely different set of cohorts, and applying it to these new cohorts is extremely predictive. So I think we have something we can predict. I'm very excited about that, even in humans. But then the question is, can we prevent? And what we are trying to do there as a next step is a collaboration with Faith Gunning and Francis Lee to use an app that they have validated in clinical populations to see its cognitive behavioral therapy app, to see if we could reduce the risk of mood disorders in a targeted way, especially in people at high risk. So I want to add to what we learned that FGF2 is a resilience factor, that FGF2 is a molecular organizer, and that in humans, initial affective status is paramount in predicting susceptibility or resilience to subsequent stress. And that sort of reminds me of the experience study that I told you with the predicting with FastTrap, because it says how you enter an experience matters in both cases. So I find that kind of an interesting resonance between the two. So, this freshman study really kind of brings home to me that we are in a difficult moment. I mean, seeing these young people at these rates of depression and anxiety is very distressing. And I hear a story from Ginny Weinberg, our research nurse, about how they're struggling, how they're feeling, and it really brings it home to me. And I have said before that I feel we are in a second pandemic, which is the mental health crisis. It's not just our data, but globally, during the first year of the pandemic, depression went up 25% worldwide. It's mind-boggling in 1 year. There is, as we know, long COVID and the pain and stress and inflammation and all of that goes along with it. There is a raging opioid crisis that interacts with the current pandemic. And there is huge societal impact on not only young women and women in general, but also people with racial disparity, poor people, healthcare workers, all kinds of populations where the statistics doesn't do it justice. It's just devastating in those communities. So an important question I want to end on is, what can science do? Science helped the other pandemic, the COVID pandemic. Can we do something? I feel like we've learned a lot, and by we, I don't mean our lab's work. I mean all of us in this room, in this meeting, in the world of neuroscience and psychiatry. We have learned a lot. And even though we don't have everything figured out, and even though it's complex, we know a lot more than we give ourselves credit for sometimes. And I think that part of what we learned is what is wrong, how to not conceptualize things like one gene for depression, or it's just a serotonin imbalance. We learned what was wrong, and we learned how to think about it better. And I feel that this knowledge is actionable, and that we should be thinking about that. We don't need to fully understand everything mechanistically. I mean, wearing a mask can save lives, even if you don't know the mechanisms of COVID. Or stopping smoking can save lives from lung cancer, even if you didn't know how it produced it. You can save lives by knowing a relationship and intervening. And the question is, can we begin to take what we know and the predictions we have and the knowledge and do something with it? And then how do we communicate the knowledge to the public? So Eric Nestler and I just edited an issue that's upcoming from PNAS on stress and depression. And we wrote an introduction about this idea of the second pandemic and what can science do. And even in the very short term, I think there are things we can do, like adopt the idea that mental health is a human right. That's WHO idea that I think is really just an important thing to just drill down in this world. That we need to articulate a coherent, scientifically grounded view of these disorders, mood, anxiety, that we can communicate. That we need to create a partnership with the public. Why aren't people wearing ribbons about all of this that's happening to mental health the way they do for cancer? We need to do better, and we need to target our message all the way up and down the line. We can't just tell some individual person, hey, you gotta be resilient. They can't. They don't know how, and it's not easy. So it needs to permeate the system all the way up and down the line, and it needs to be systemic and serious and evidence-based, and move forward from there. So I feel like science can do a lot, and I feel like the time is now. So I want to end with this note that we can provide hope, and I hope that we will. Thank you.
Thank you so much, Dr. Akil. We are now going to have a time for questions, and I'd like to introduce the co-moderator, Dr. Eric Nestler, that you just heard about, who is Professor of Neuroscience at the Icahn School of Medicine at Mount Sinai, to ask the first question.
Thank you, Huda, and congratulations on winning the Gruber Prize and on giving us such an outstanding lecture. I also wanted to offer a personal note and thank you for being such a wonderful mentor and colleague to me and to so many other society members over the years. So a heartfelt congratulations.
Thank you. Thank you. Thank you.
The Michigan freshman study data are chilling, and we all knew that before the pandemic, the incidence of stress disorders among young people was increasing, and then since the pandemic, it has skyrocketed. And the statistic you showed of close to half of college-aged women having a diagnosable syndrome of depression or anxiety should be a national call to arms. So if you could expand still further on what you think we could do as neuroscientists and as members of our society at large on addressing this second pandemic, please.
Yeah, I think, thank you, Eric, first for the kind remarks and also for the question. I think that first we need to form partnerships within our disciplines, like with psychiatrists, for example, and with healthcare workers of various kinds. And we need to get on the same page about when to intervene, how to intervene, but also all the modalities. So this idea of using an app, for example, is not something that could have been done 10 years ago, but one can do it now, and it can be evidence-based. Does it work? But does it work for some people? Does this approach, this psychiatric approach work for this group versus that group? As we also know, there are some brand new treatments with very different bases that can be used. There are, different paths to depression. So some of them are, situational. Some of them are metabolic. My daughter is here and she works on that. Some of them are related to inflammation. So instead of trying to bundle everything into one category, we need to parse it enough to be able to improve this prediction and relate the cause to the intervention. And there is AI and computational approaches and brain stimulation. There is a ton, and people don't know about it, and they don't know when it's appropriate. We don't have paths the way some other, like if this, then that, there is the sequence. We could begin to develop those more explicitly. Maybe some people know it, in academic fields, but the public doesn't know where to look or how to find it. So we need proof of concept that all of this is doable, and that's not one group who can do that. I think as a society, as a community, we need to do it.
Thank you. There's a question from the audience around the inheritance of temperament. Is there any gender differences? You've shown some in the animal models. Is there a skew for transmission from paternal or maternal genomes, as far as you can tell?
I don't know. It's a great question, and honestly, I should know, but I don't know. So the data in the human are still patchy. They're not like complete or beautiful. In our animals, we see it both in males and females, more or less equally, although, as we drill down more, so all these studies on temperament, we do have both males and females by definition. As we drill down more, we can see if we can look at this question in a more systematic way. I would not be surprised that there are interactions, if not, an epigenetic, and the way that different genes come together and different pathways, and maybe that even the data analysts like Megan would know, but I don't know. But it's a great question, thank you. I will try and learn more about it.
Huda, several audience members asked about bipolar disorder, to which you referred at several points during your lecture. Can you say a bit more about how your LR and HR rats might provide insight into features of bipolar disorder, or whether your studies on the glucocorticoid receptor might contribute?
So to me, the best evidence is from the GR overexpressing animal. And what was very notable is a whole series of genes, especially calcium-related genes, that have popped up in common in bipolar disorder as very high hits, and also in our GROV animals. And Joanna Spencer-Segal has also looked at calcium responses in the brain in the hippocampus of our GROV animal, and they respond differently with calcium imaging to their environment. They encode the environment differently, they react differently, and she has a paper on this. So I think something is changing calcium in particular pathways, and as well as glutamatergic function. And I think that might be a useful insight in bipolar, in this one dimension of bipolar, which is that this ability to flip around in some cases with great ease, suggesting instability of a connection or a positive feedback loop, which is what I like about the circuit, is glutamate to glutamate, and you could see how it can go haywire if not well controlled. So there is a lot of work to be done on that, but I do think that it might turn out to be kind of an unusual angle. So I think I want to do single cell transcriptomics on the GROV animals and compare it to single cell transcriptomics in bipolar illness and see if we can see things in common and use that as a way to target particular genes and learn more.
Given your findings around resilience and taking together the human as well as the mouse data, how are you thinking about further studying the potential for critical periods for the development of developmental critical periods for the development of resilience?
Yeah, I mean, there is something I used to always think about early life, and it's obviously very important. And if you grow up in the stress literature, you think about that a lot. And the GROV animal is an example of how much happens very early on. But the adolescent period is something that I've started to think about much more lately. And it happened for a very weird, interesting reason. We were doing intervention studies, and different people in the lab would come and on these HRLR animals would come and tell me something really weird is happening. We are flipping these animals around, like they didn't know about each other. So somebody is doing stress, and suddenly the low responder animals are acting, if it's mild stress, somewhat more resilient than the high responder animals, or sensitization to drugs or something like that. And I started to, I thought that we were going to be hitting them and making them worse, but sometimes we were rewriting the program. And I think this is a time, and maybe this is where understanding the gonadal hormone is going to be really important, is at that point, that it reopens the chromatin organization and it allows more epigenetic changes. And so we will be able to see that with like ATAC-seq and looking at how the transcriptional profile changes during adolescence and with interventions during adolescence. So I think from a scientific level, this is a time of great plasticity, and it's a very interesting time where you can rewrite the way the genetics are playing out in the level of the brain. And from a more societal point of view, obviously it's a very important thing to communicate about how we treat our adolescents, how we think about our adolescents, how critical these periods are, that we need to provide them with the resilience and the support. And not to say, well, he's always that way, or she's always been sensitive or difficult or whatever. That this is a time where you can relearn a lot, reorganize a lot, and give them tools. So I truly believe that this is one of the lessons that can be nicely communicated to the public.
Huda, back to the freshman study. We received several questions from the audience about more details of the study.
Yeah.
So one of the questions, for example, is whether the prevalence of depression and anxiety might vary by the student's major. One of the audience members, it's interesting, one of the audience members suggested that perhaps the problems might be greater in STEM because of the more difficult curriculum.
Yeah.
Others posited that perhaps STEM students do better because they're more resilient.
Yeah, so we have not done this, although we have started to talk about things like that, or also if they change majors. There's also, but but relevant to your question is also a selection bias, and in particular, how I think about the people who are high genetic risk but did not succumb to depression. So I feel like if you're walking around with some family history and a high genetic risk for depression, and you are able to get into University of Michigan, which is pretty competitive, you already know a thing or two about resilience. And so what I don't know is if we have a somewhat biased sample, because we're picking a higher proportion of psychologically resilient kids than would be in the population at large. But I think now we have numbers that are large enough that we can go in time and drill down on some of these questions of other predictors, other than the affect score, which is relatively new for us, and we were excited to see it. Are there other predictors that are very easily measured, like your major or any other characteristics? I'm also interested in the endocrine profile and how it's predictive, but that's a whole other story. But yeah, I think that there is a lot to learn from these kids. Sleep is also what one can learn more from that. There's lots to learn.
Well, there's a question that we have here, which I think will be important for a lot of people in the audience. What advice do you have for young investigators who aspire to be like you?
You don't want to be like me. Here I am at my late age. I don't even know how to stop, so you know. But what I would say is probably the thing that helps the most is to have a sense of proportion. I'm from Syria, and for a while I was writing grants on a topic I cared about, but it was probably like overly ambitious, and they weren't coming, and people were asking me if I was really upset. But I had relatives during a civil war that were dying. And that always gave me this sense that, the world is falling somewhere else. I just didn't get a grant. It's not the end of the world. So I feel sometimes that we love science, but we make it so powerful that we are totally overwhelmed by it. And having some sense of proportion and some sense that if you have good ideas and enough people publish your papers and to discuss with you, that you probably have something, and you just need to figure out a way around it. That I think has helped me last over 50 years in this field. So maybe that's helpful to you.
Thank you, Dr. Akil, for a fantastic lecture, and congratulations again on the Gruber Prize. Thank you.